Okay. Hi, everyone. Let's discuss a paper right after reading it. In this journal club, we picked up a paper 2 hours ago and then started reading it. From now, we are going to discuss the paper. Today, we have two people, Changshan and me. Changshan, can you introduce yourself?
Yes. Hello, everyone. My name is Qian Chen, and I am currently a PhD student at Monash University, Australia.
Okay, thank you, Changshan. And I am Yo Takashima. I'm doing research engineering in Japan. And today we are going to discuss the paper, Geometric Constraints on Human Brain Functions, published in Nature 2023, this year, April. So just 1 month ago. The authors are James Ban et al., and all of them from Monash University, I guess, or at least the first and the last authors from Monash MBI.
Yeah, the
So
same thing as our lab.
yeah, so
Congratulations.
yeah, it was really interesting paper. But anyway, so I'll first give you a quick summary of this paper, and then we're gonna start discussions. Right. Okay, so the paper, as the title says, talking about geometry, or simply just the shape of brain. So in physical world, the shapes determines its function in various things, right? For example, like a drum, they discussed in the discussion, they talked in the introduction. So the drum shape, the shape determines its sounds, right? Likewise, their idea is that maybe the shape of brain determine the functions, behavior of the brain. That's the idea. And of course, this idea is not that, rare in a sense, because people are talking about anatomy of brain and try to connect the function of brain with each brain regions. And the classical approach is to think about the connectivity, specifically like a axonal connectivity across cortical regions. And this approach is called a connectome approach. So they try to, the starting point, the motivation is that there are some population of neurons specialized for specific functions, and these functions are connected across cortical areas, and they have, as a whole, have a unique functions. That's a kind of classical connectome approach. But in this paper, they not propose, but they try to test an alternative approach, which is called neural field theories. So the idea is that instead of thinking about the brain as a component or each independent sub-regions, rather this approach thinks the brain as a continuous some object in a sense. And this, continuous sheet of neural tissues, we have this sheet, and then through this sheet, the waves or activity propagates. So instead of thinking about each component, each brain region, we first take the brain as a whole first, and then try to see the spatial pattern of activity of the whole brain. So to test this idea, what they did is to analyze fMRI activity data from various conditions. And they use the computational model or just, implement the two approach, connectome approach and neural field theory approach, and compare the performance of reconstructions. So the both connectome and neural field theory can or kind of decompose one brain states into unique pattern of activity, spatial pattern of activity. And then this, in a sense, by summing up all of these unique patterns, spatial patterns, we can have one brain states and in different task conditions. And when they compare the two approach, connectome and neural field theory, the performance of reconstruction is better for neural field theory than connectome approach. And that's the first result. And also they try to see how the neural field theory approach behave in different task conditions. So in order to do that, first of all, they found that spatial pattern with long spatial frequency dominate rather than just a small, tiny, spatial patterns. That's the main another finding from the first analysis, I think. And then in the second analysis, they confirmed that in various task conditions. And then they found that the reconstruction performance drastically decreased when they remove the long spatial frequency patterns when they reconstruct the brain activity than removing the high spatial frequency patterns. So this indicates that the high spatial are not important, but the low spatial frequency pattern is dominant and may be more important to explain or just to reconstruct the brain activities. That's another analysis for the findings of this paper. And also, so far I've talked only about the spatial pattern, but they also check the temporal dynamics of these patterns, spatial patterns. And they found that in the neural field theory approach, they could found some, brain dynamics, which is empirically observed, such as visual hierarchy from lower visual area to higher visual area. So the visual area has two pathways, dorsal and ventral. And from the analysis, they observed that the activity propagate from lower to higher area along these two pathways. That's another, temporal dynamics or findings in temporal dynamics. They also confirm that this kind of, or the approach can be applicable to non-neocortical activity, such as thalamus, and they found that the approach is generalizable to non-cortical area. I think that's basically it from me. Yeah.
Okay. Yeah, thank you very much for your summary. You mentioned a lot of details that I did not pay a lot of attention to. I think I'll add one concept to the background of the the paper. So here, as you mentioned, the authors wanted to test two theories, which is whether the kind of complex connections between these local regions across the brain, or just the shape, geometric shape of the brain explains which one explains the brain activities better. So the key difference between the two theories is that whether their model take into account the physical, geometry of the brain, in a sense. The discrete connectocom view, which we previously said was about the complex connection between brain regions, do not really consider the shape of the brain itself. So I don't fully get what it means they have like an abstract sense of the shape of the brain, but I think what they're trying to say is if two regions talk to each other very frequently, then they are kind of considered close to each other. Even if they're actually physically very far away in a sense. But the neural field theory does take into the geometry into account in the sense that the cortical interaction is specified by some kind of activity or like some kind of traveling waves that decline exponentially with distance, physical distance. So if the two regions are really far away from each other, then their wave dynamics may be different from the two regions that are really close to each other. So they need to take into the take physical distance into account. So that's the key difference. And did you mention the geometry constraint brain function part?
Brain functions part.
Oh, waves bridging geometry and function, sorry.
So that part, to be honest, I could not understand because my understanding is that when they say functions, I interpret that it's dynamics. Brain dynamics, activity of brain dynamics. So I don't think they mentioned like, okay, we can explain like this, I don't know, function or specific process. Like, I don't know.
So explain dynamics with dynamics.
Yeah.
So
they don't talk about like functions. I don't know.
I see. My understanding is that this section, based on the previously, they already established results of brain geometry, explains brain activities very well. And now they want to understand how does the shape of the brain, which is geometry, shapes the function, like through what mechanism. And they use they borrow or like they use an idea in physics where the wave dynamics of a physical system constrains its function. So it's just like the sound of violin is characterized by its sound waves, and it was the shape of the violin that determines what the sound waves is like, and that segregates it from the sound of piano or something like that. And likewise, they are thinking maybe we can compute this kind of sound waves, which is the wave dynamics of the brain based on its shape, and to use that kind of computed wave to see whether the waves explain the brain activities. And the results was that the wave model explains the neural data very well compared to some kind of traditional, very complex model.
That part I'm not sure, because I think that part corresponds to figure 4, I think. And
Oh.
this part I could not understand, to be honest. But the B, subplot B, compare two models, and from these figures, I don't think the one is, perform clearly better than the other. That
Oh.
is my impression. So what they say is maybe more, they need less not assumptions, but maybe less consideration. So in a sense, more unified when we use the neural field theory, I think. Because they repeatedly says that this, parsimoniously, which is, I think, well used in mathematics, meaning that we can explain one single things with few explanation, in a sense, or few equation, much simpler.
Yeah, so they also said this wave model shows comparable or superior performance in the reconstruction of empirical data. So they probably are not saying this is way better than the existing models. What they're saying is, it also explains the existing data very well, and it's a simpler model, so we should adopt the same simpler model.
Yeah, I think that's the point. One, important point of this paper that is discussed in the discussion part. This approach is much simpler and than the classical one, so why not use that kind of argument. But anyway, so thank you for adding some missing parts, Qianshen. And so shall we move on to discussion, clarification question part? Do you have any questions?
Yeah. So not questions regarding the paper content itself, but more like a thoughts. So I was thinking how we can use this to think about the neural correlates of consciousness. So one way to think about it is, can we use this to evaluate what kind of system can generate consciousness? So maybe human brains or brains of some animals, they can be conscious because they satisfy certain geometric requirements.
I see. So you're saying, like, for example, we think of ball-shaped brain, like just a circle, versus triangle, versus much more complicated, like brain. Which shape more, is more likely to have, consciousness?
Or something like that. Maybe the geometric requirement is definitely something much more complicated than just it's this shape but not square or triangle. But so if we have to satisfy these requirements, then stimulating a consciousness in some flat chips is probably very difficult.
Yeah.
But we don't know at this point, but it's just a thought. But even so, I feel like we cannot just discard the investigation of the complex interconnection between the brain. So you can imagine, for contents of consciousness, you can have different experience without major differences in the shape of the brain, right? It's like, is there maybe I don't fully understand the so-called geometry of the brain this paper particularly means, but for example, colorblind people and neurotypical people, do they really have different brain geometry to have different color experiences? Maybe not.
So yeah, I think the brain geometry, my understanding is just a spatial pattern of activity. So because I think they use the standardized brain structure, right? I think in fMRI study, it's, I think almost every studies do that, but each single participant have different brain anatomy, right? The structures. So in order to compare the result across participants, we need to have a unified brain structure. So we try to map these, each participant activity into the same brain structures in a sense. So they used this, in a sense, the same brain structures for each participant. And then on that, they plot the activity patterns, spatial patterns. For example, the frontal activate more, posterior not activated. That's pattern 1. And the pattern 2 is the opposite. And the pattern 3 is like the top area activate, but the bottom does not activate, something like that. And the combination of these spatial activity is, I think, geometry, what they mean. I think. And of course here, even though we ignore the across participant difference, the brain has its own, or the human brain has similar structure across participants, right? So this, I don't know how to call this, but like this kind of shape. Like this shape. Yeah. And then this shape is also important, and this also somehow means geometry, I think. The combination of this and the spatial pattern of activity is the geometry in this paper, I think.
So yeah, I think probably the key here is like when they reconstruct these activities, what kind of things they take into account. So the one that takes into account of distance between regions is the one that take into account of geometry. So it's like distance. And then the connectocom one does not. So that's like the difference.
Yeah, so you mentioned that, we still need to think about the intercortical, connectivity, axonal connectivity. And while I read, I was reading this paper, I also had the same feeling because, and also in the discussion part of this paper, they mentioned about that. But maybe I think this axonal connectivity requires much more high resolution, data. I think. Because again, so in standard fMRI study, as far as I know, they try to standardize each different brain from different participants into one. So in a sense, localized activity for each participant is kind of average and broad, right? So that reduce the spatial resolution in a sense. And also they said that, they use human connectome data. But it doesn't mean that, that connectome data is obtained from single participant. Rather, it's averaged across 1,000, I don't know how many people, but, many participants. And then estimate, okay, for, somehow standard normal people will have this kind of connectivity from here to here, axonal connectivity.
I think the Connect to Come project provides data from individual and you decide what you want to do.
Okay.
Yeah, they
I see.
But and as far as I know, I do remember reading in another review paper about how averaging may not be a good idea because the resulting activity pattern could be from a highly variant activity pattern across individuals, and it's not representative of any single one of them.
Yeah, so that's, kind of another not concern, but the comment I heard, because when they say, long frequency or long wave, patterns, they're talking like almost like 6 millimeters, so 6 centimeter is the order is centimeter always, and 6, 4, 3 centimeter, right? And then I don't know how each participant has, or how the brain anatomy is different across participants. But the reason why they see only above 3 centimeter, spatial frequency is that they did not consider each single participant as separate participant, rather average across, and that's kind of cancel out the low frequency component, maybe. But again, I haven't read the method part in detail, so I might be wrong.
And also they mentioned that they analyze data from many different experiments, like 1,000 or something experiments. And each experiment could have, let's say, 20 participants. So it's probably computationally impossible to do analysis on each individual.
So I guess, yeah, my guess is that in order to have these spatial patterns, eigenmodes, what they call, they use just pool all participant data in, and then have one spatial pattern. That's my guess. But again, I didn't read the method part, so I might be wrong. I need to read that part. Yeah, anyway, so basically these are the comment I just like comments. Other thing is that, yeah, but
yeah, I don't have other comments or questions.
Okay, so other thing is that maybe, so the previous comment kind of, criticism on this paper, right? So the reason why, I mean, my comment was not the criticism, strong criticism, but rather the reason why they didn't see the high spatial frequency result is, or the reason why they see better performance from this neural field theory than the connectome is that the averaging or low resolution data. But another point is that maybe, so when this is again just my guess, but when we try to understand the brain, systems behavior, how the brain behave as a whole, and then when we measure activity at the scale of fMRI, then we might not need to be careful, or we may not be consider such fine, detailed spatial resolutions. Or, I mean, like the connectome axonal connectivity. And the reason why is that this is just an analogy, but when we try to understand the behavior of objects, for example, like a moving soccer ball, like from right to left, we of course do not think about how each atom or molecules constitute the ball behave to understand the whole behavior, right? So in a sense, we can say the same thing to the brain behavior. So the movement of ball corresponds to the behavior at the system, the whole brain level, and the behavior of atom maybe corresponds to like a single neuron behavior or axonal connectivity. I'm not sure about this because the scale is too different, right? The atom is too small well, compared to the soccer ball, but the brain structure, the brain and axonal connectivity is not that different, I think, in terms of scale. So we can't say, or we can't use this analogy, but this this is just my understanding, but this paper kind of saying this point, I feel. I feel, but yeah, my impression. So because in the discussion they said that to explain this level of activity, we don't need to consider connectome. We can just simply think about, okay, first the activity propagate like a wave in a water pond. And also we have this structure. So if the activity or the brain region around this area activate, then that will propagate like beyond, and that's it. And then that whole brain activity can be explained by the combination of these. That's it. We don't need to think about how each region is connected or something like that. That's the idea of this paper, I think.
Yeah, I agree. But as for your previous comment on the criticism, like on the, how the long wavelength explains the activities much better than the short wavelength. And it could be a result of averaging. I think I need to find. I think they did a lot of extended analysis to verify this point and to exclude a lot of additional alternative explanations in the supplementary material. So
okay. Yeah.
Yeah, so maybe they have answered that question there.
I see. Yeah, I think this paper is published in Nature, so almost I think at least I came up that pretty soon. So I guess someone already asked them about that. So they should have something to defend this idea. So yeah, I may need to read the method part. Yeah, because method part itself, I don't know how long.
The methods is like almost the majority of the paper.
Yeah.
Of like the entire document, let's say.
Yeah. So
4-page, and then there are supplementary materials.
Yeah. So, but yeah, this paper was interesting.
Yeah.
Yeah. Yeah. And yeah, I think that's all from me. Uh, um, yeah, I think that's all from me. And you said you don't have any other questions, right? Okay, so shall we finish now? Or let's finish now. Okay.
Yep.
Then
All right.
see you next week. Or Chachen, you can ask these questions directly to the author because you're in the
Yeah.
same building. Yeah.
Yeah.